Okay, hello again, folks. So we're bouncing straight on into polypeptide synthesis part 2. So this is the bit on translation. So if you haven't watched part 1 yet, go back and have a look at transcription because we're going to pick up basically where we left off with translation. Now it's quite a simple process, this one. It doesn't have any other level of weird complexity like splicing and capping and all that kind of stuff that the last one did, so it should be relatively straightforward compared to that. But I left you last time on a little teaser. Now here's the answer, the terminal 3' tail teaser. So I asked you, if a mutation occurred which knocked out the stop codon, which destroyed the stop codon, what amino acid would the protein be rich in? Okay, and the answer to that is lysine. If you got to that, well done. Reason being, if we look down here, if the mutation knocks out our stop codon, okay, then our ribosome, which will start here and run along, won't know to stop, okay, and it will just carry on after the end of the gene, okay, into what's called the 3' untranslated region, and then eventually into the poly A tail, which, as we know, is just a lot of AAA, okay. So that AAA, 3 A's translates to lysine. Okay, so if it's running into there, it's just going to add an awful lot of lysine to the end of your now probably dysfunctional protein. Okay, now that kind of question required you to bring together a knowledge of the genetic code, a bit of logical thinking about what the point of the stop codon is, and a knowledge of the structure of RNA from last lesson. So that's a high level synthesis question. So if you can do that kind of thing, then you might be looking forward to going off and doing some super high level course, because that could be the kind of starter question for some sort of high level interview that you might do down the line. Okay, right, let's move on to today's material then. So this is the DNA and protein synthesis. As you can see, look, translation is the production of polypeptides from the sequence of codons carried by mRNA, the role of ribosomes, tRNA and ATP. So that's very important that you know what those do, but again, it's equally important that we keep context. So last time we talked about the detail of what happens here in stage 1, transcription. Today, here, translation. So what happens with this now mature RNA when it actually binds to the ribosome? What goes on there? Okay, so let's dive straight in then. Okay, so in terms of translation, you will know what the rough endoplasmic reticulum is. In a eukaryote, that's where this is all taking place. In fact, the reason the rough endoplasmic reticulum, you can see it here, is rough is because it's covered in ribosomes. In a prokaryote, this is just all happening, flopping around in the cytoplasm. Okay, so we're building a polypeptide chain from amino acids, and it's guided by the sequence of codons on the mRNA, as you've already done in the genetic code lesson. And the structures involved are all things you should know that you've done in other lessons as well. Messenger RNA, the mRNA, transfer RNA, the tRNA, both in the RNA structure lesson, and then you've got a ribosome and an amino acids there to build the protein. Okay, so those are our ingredients. And it's broadly broken down into 3 stages. You've got the initiation stage, so how does it start? You've got the elongation stage, so how does the protein get longer? And then you've got the termination stage, which is how does it stop? So we're going to go through those one at a time. Okay, so here we have nice, simple. It's a little bit dull in colouring, but I think it gets the point across nicely. Okay, so I like these diagrams because they're quite nice and simple. So what happens on the RNA is you get a place called the initiation codon. Now, we mentioned this when we did the genetic code. It's always AUG. Okay, and what happens is the mRNA, a tRNA, and the first amino acid, and the 2 ribosomal subunits join together around that initiation codon. Okay, so they attach to the AUG. The small subunit comes on first, then the tRNA, then the large subunit. Okay, and then we're off. Okay, they're upstream of the initiation codon, and that's called the P site. Okay, in the ribosome, you don't need to know the different sites in the ribosome, but that's what that is. So the ribosome sort of moves along, so we end up with this site here. Okay, vacant. Right, so in comes our tRNA with our methionine in there, and initiation has begun. Okay, so we've now got a vacant site. It doesn't take a genius to work out what's going to happen next. Okay, now then we're into the stage of elongation. Okay, so what then happens? Look, amino acids are added as the ribosome moves along the mRNA. The tRNA binds and a peptide bond forms. So here we've got our second tRNA forming into the ribosome and a peptide bond form. As you can see, look, there's a cut made in there, so the ribosome does that, cuts the tRNA off, and we move along. Okay, and tRNA at the P site here is released, and then this A site here, as it's called, when the ribosome moves along, becomes the P site, and we're left with the A site vacant, and in comes, in this case, cysteine, tRNA, and the same thing happens again. Okay, so very straightforward, very simple process. Once you've found that initiation codon, next tRNA comes in. Now remember we said tRNAs are like adapter molecules. They've got the anticodon on one end and they've got the amino acid on the other. So they'll bind on to your mRNA, okay, and drag that correct amino acid in. Turns out they don't actually know anything like this. If you were to biochemically add the wrong amino acid in, they would drag the wrong amino acid in. There's nothing magical going on here. There's just a series of enzymes that attach the right amino acid to the right tRNA, okay? You can also guess what's going to happen here between this leucine and this cysteine here. You're going to get another peptide bond forming, okay? And as you should know, that's a condensation reaction, right? That's going to produce water, and off we go. Now, all this process here, right, running along to move the amino acid, to bring things in, takes ATP, it takes energy, right? So cells that are involved in a lot of protein synthesis require a lot of ATP. So if you see a cell down the microscope that's got an awful lot of rough endoplasmic reticulum, it's likely also to have an awful lot of mitochondria as well to produce that ATP. Okay, so this runs on, and amino acids can be polypeptides can be thousands and thousands of amino acids long until you hit a stop codon. Now, if you were looking carefully back in the genetic code lesson, you will have seen that there are 3 codons that don't have a tRNA associated with them. Okay, they are the stop codons, right? And what happens when you get to that point? There is no tRNA to go in there, so the ribosome cannot continue to function. A thing called a release factor moves in and basically causes the ribosome to fall apart. Okay, so it falls apart after cleaving the last tRNA off, done, and releasing that polypeptide. Now, if that's in a eukaryote, that would be into the rough endoplasmic reticulum. And then we're back into the cell biology bit, that the rough endoplasmic reticulum is then involved in folding up the protein, the secondary structure, tertiary structure, so on and so forth. That then gets moved into vesicles to the Golgi and distributed to where it's supposed to go. If it's in a prokaryote, then that's just released into the cytoplasm, because it's all happening just in the cytoplasm. Very slapdash, right? Which is obviously very quick, but it's also very inaccurate. So that's our stop codon, and that gets released, right? So that's the basic process of translation. So I told you it was relatively simple, right? There are some additional complications. So this is our kind of stage, our level up in this lesson, which is post-translational modification, okay? So once translated, the polypeptide chain can obviously be changed, okay? It can be modified, and that's obviously for very good reasons in a lot of cases, okay? So you might recognize this little fellow we're going to get to in a while, okay? Here are some of the things that can happen. Right, you can get the removal of methionine, okay? The start codon for each gene, right? So is methionine. In some cases you don't need that, so that gets taken off, okay? It doesn't want to be methionine as the first one. You could also get the addition of functional groups. So you could add phosphate groups to your protein. You could add acetate to your protein. In the case of hemoglobin, or anything with a prosthetic heme group, then obviously a prosthetic group of any sort, then you're adding things like iron. In this case, if you're a crab, you've got hemocyanin, you're adding copper. Right? That's why they've got blue blood and you've got red. Interesting fact. Structural changes, of course. Okay, so the addition of things like disulfide bridges, or just trimming the protein. And a really good example of that is insulin. So insulin isn't manufactured in its functional form. It doesn't instantly become working insulin, the hormone. What tends to happen is you've got pre-proinsulin, which is the chain, the primary structure. That then folds up and starts to form some disulfide bonds, disulfide bridges. Okay, we call that proinsulin. But that then has to have a further stage done to it where it actually gets trimmed, it gets cut to create this final shape over here, which is functional insulin. And as you know, this one here, look, is hemoglobin. And hemoglobin is made of 4 polypeptide chains, so it's a quaternary structure that have to interact together, and each one has that prosthetic heme group made with an iron in its centre. Those are all what we call post-translational modifications. Okay, so after the translation's happened, that's when you get into other parts of the AS level spec, when we start talking about secondary, tertiary, and quaternary structures, and the addition and cleavage and chopping and trimming of various things. Now, this chopping of protein is really, really important, because if you think about what you know about digestion, protease enzymes are produced in the stomach, right, in the lining of the stomach and in the pancreas, okay? But of course, most of your cells are made of protein. So do you want an active protease, a very broad spectrum active protease, running riot inside your cells? Well, no, of course you don't. So most stomach proteases are manufactured in an inactive form, right? And then it's only when they get into the pH, the low pH conditions of the stomach, that causes hydrolysis, cleavage, cutting of the protein, makes their active site into its fully functional form, which then is able to chop up proteins in your stomach. Very, very clever and important use of post-translational modification. So the basic level is say what happens during initiation, elongation and termination, and then as our step up today, we've mentioned post-translational modification as well. Of course, all of this is a very energy-hungry sort of project, so you need to have lots of mitochondria around wherever ribosomes are working. Okay? I told you it was a short one, so I'm going to end with a little game, a little challenge. See if you can do this one. This is a pause the video, see if you can get the answers. We're going to have a look at structures and processes involved in protein synthesis. So what I want you to do, pause the video now, and for A, B, C, D, E, F, G, H, I, J, so on, they are all things. They're all structures. They're not translation or anything like that. They are all things. So pause the video now, get an answer to what you think those are, and then we'll get the answers in a moment. Okay, good. So let's get some answers. Right. So if we come down here, look, A is our DNA molecule, B are our free nucleotides, C is RNA polymerase, D is an mRNA molecule, E is the nuclear envelope, F is nuclear pores, G is an unloaded tRNA, H are the free amino acids that are about to be loaded on the tRNA, I is the ribosome, J is the polypeptide chain emerging from the ribosome. Okay? Okay, so that's structures. I hope you got all of those right. Let's have a look at processes now. So now we're going to do the same with 1, 2, 3, 4, 5, 6, 7 and 8. And these are things that are happening. So they're not structures, they are processes. So pause the video now. What is happening at 1, 2, 3, 4, 5, 6, 7 and 8? Okay, great. I hope you've done that now. So let's have a look at some answers. Well, number 1 is the unwinding breaking of hydrogen bonds with DNA helicase. Number 2 is the adding of our complementary RNA nucleotides to create mRNA. Number 3 is the rewinding of the DNA molecule. Number 4 is the mRNA moving into the cytoplasm. Number 5 is the tRNA with the amino acid being drawn into the ribosome by complementary base pairing. Number 6 is the amino acid added to the growing polypeptide. That obviously uses ATP, and obviously various other bits are not shown in that picture. Number 7 is the unloaded tRNA leaving the ribosome to be recycled. And number 8 is amino acid activation. So this specific amino acid being joined onto a specific tRNA by a very specific family of enzymes that we don't go into at A-level standard. Okay, so I hope you got all those right. That is us. That's the entirety of transcription translation and is the end of this topic. Any questions, do get in touch. If you feel like you want to go a bit more advanced, do go and look at the gene expression topic from year 13, from the full A level, which is a bit more higher level, exactly how we know which genes to switch on, when and why. Okay, many thanks, and I'll see you soon. Bye-bye.